I'm leading several programs of the modulation of food quality to modify the gut microbiota and the cardiometabolic health. And I work on several research programs on the modulation of food quality in order to alter gut microbiota and cardiometabolic health. In my lab, we are performing clinical investigation. We work on human beings. We have some nutritional intervention in order to see whether some food quality and some different ingredients could prevent or help to manage cardiometabolic risk. This topic is important because we want to work on the modulation of food and how the ingredients and the food we will ingest will modify the gut microbiota function and composition and will help to prevent or to modify metabolic parameters such as lipid profile or glycemic response, for example. So with this new project we are leading now with Lallemand team, we want to work on existing lactobacillus, and we want to see whether this lactobacillus will impact beneficially the lipid profile. So with this new project we are launching with Lallemand, we want to investigate whether this kind of a lactobacillus can beneficially impact the glycemic profile in response to a high-fat, high-lipid meal. And this will be performed before and after several months of ingestion of the lactobacillus. And we will see whether it will help the people to handle this nutritional challenge that is for us early marker of the deterioration of a cardiometabolic profile. It's a clinical trial that will be performed in men and women preventing with a high cardiometabolic risk, but not only already under medication. And we want to see whether the ingestion of some lactobacillus for several months will help to limit the alteration of their cardiometabolic profile and the appearance of further cardiometabolic alterations. We work a lot in my lab on the modulation of gut microbiota and more specifically for several years on prebiotics, but we wanted also to investigate other ways of modulating the gut microbiota, working on probiotics now. The originality of our model is we want not to work on fasting samples, for example of blood or feces, but we want to work on postprandial metabolism. Why? Because a human being spends more than 3/3 of his life in a postprandial state, and we think that it's a very early marker of deterioration of the cardiometabolic profile, and that will help to decipher early the potential risk of developing further alteration. Up to now, we cleared all the E6 steps last year, and we are on an ongoing recruitment with almost half expected population recruited. We launched this collaboration a few years ago, starting discussing about the scientific objectives of the project together with the R&D team, and we have a very good relationship because we've been working together initially as collaborators in the lab and now in this collaborative aspect with industrial and academic profiling. Our collaboration with Lallemand is based on complementary expertise. We work a lot in clinical trials, nutrition, metabolism, and they bring all the expertise regarding probiotics and microbiota. This will be a first clinical trial that will go that far into the metabolic phenotyping on the impact of this lactobacillus, and it will help us to understand how this kind of probiotic can impact beneficially the cardiometabolic profile. This study will be the second validation study that will investigate the impact of this lactobacillus, and the first one to deeply investigate the metabolic profiling based on postprandial metabolism and multiomics. We will be very happy to show you in 1 year the first result of this clinical trial.